I'm Rita. Our short presentation is about gene expression and protein folding in relation to Tay-Sachs disease. The central dogma of biology is the concept or principle that DNA is a coding sequence that is transcribed into RNA, which in turn is translated into proteins.
This process works by 1. Transcription. RNA molecule is produced by RNA transcription before the synthesis of protein begins. RNA polymerase uses a strand of double helix DNA as a template to synthesize messenger RNA or mRNA. The mRNA travels from the nucleus to the cytoplasm. This is the time when the messenger RNA can go through various types of maturation, such as splicing. Non-coding sequences are abolished during this type of maturation. A codon of DNA or RNA is a sequence of 3 adjacent nucleotides which codes for a specific amino acid. For example, according to this table, for glycine, the first and the second letter are always going to be a G. For example, a first letter and the second letter are always going to be a G. And the third letter will be a random nucleotide such as U. So it can be a GGU.
Step 2. Translation. The tRNA transports the mRNA to the ribosome where it binds to the start codon. The elongation phase begins of protein synthesis. This is where the tRNA enters the A site of the ribosome and shifts to the P site until another tRNA enters the A site. When this happens, RNA forms a peptide bond to each amino acid that is attached to the tRNAs. The first tRNA shifts to the E site where it is ejected and recycled. This process happens until it reaches a stop codon, terminating the translation process and releasing the complete polypeptide from the ribosome.
Tay-Sachs disease, a rare inherited disorder that progressively destroys brain and spinal cord nerve cells. Most common forms is in infants where they lose motor skills. This later progresses and the child will experience seizures, vision and hearing loss and paralysis. It is less severe in childhood, adolescence and adulthood in comparison to infants. Signs and symptoms include problem with movements due to muscle weakness, mental illness, and speech difficulty. Disease appears between the age of 3 to 6 months in infants. Children with severe forms of this disease usually only live into their early childhood. Mutations in this gene can cause Tay-Sachs disease. This is the map of chromosome 15. The hexa gene is located in between base pair 72,635,777 to base pair 72,668,519 on chromosome 15.
The hexa A gene, which is mutated, is the cause of this disease. It instructs a part of an enzyme called beta-hexoaminidase A, which is important for brain and spinal cord functioning. The enzyme is located in lysosomes that break down toxic substances and act as recycling centers. Beta-hexoaminidase A in lysosome breaks down a fatty substance called GM2 ganglioside. Mutations in the gene affect the activity of this enzyme, and GM2 ganglioside therefore cannot be broken down. However, it accumulates to toxic levels and destroys the neurons in the brain and the spinal cord, causing Tay-Sachs disease. Tay-Sachs disease affects the function of a lysosomal enzyme and involves the buildup of GM2 gangliosides. So this disease is also known as a lysosomal storage disorder or a GM2 gangliosidosis.
So, how do people inherit Tay-Sachs disease? It shows an autosomal recessive pattern where the parents of an individual with an autosomal recessive condition each carry one copy of the mutated gene, which is represented by little r's in both parents. But they typically do not show any signs or symptoms of the condition. However, the individual with the homozygous recessive allele, which is 2 little r's, will be affected from the disease. This means that there is 1 in 4 chances of inheriting the disease from parents who carry the recessive allele.
The article we looked at is called The Inhibition of Endoplasmic Reticulum Associated Degradation Rescues Native Folding in the Loss of Function Protein Misfolding Diseases, such as Tay-Sachs disease. It was published in the Journal of Biological Chemistry on October the 17th, 2011. It's a very recent article. This article expresses that in Tay-Sachs disease, mutations occur in the hexaminidase gene by the endoplasmic reticulum associated degradation of mutated, unstable lysosomal enzymes. One of the most common hexaminidase mutations is the G2069S substitution that undermines the native folding of the hexa-A alpha subunit rapidly processed by the ERAD, which also impairs the trafficking of secretory proteins. In this study, they have exploited a process called endoplasmic reticulum degradation, which is a mechanism by which only correctly folded proteins are transported to their final destination and misfolded proteins are degraded. It is a selection process. They have achieved to inhibit this process by cell treatment with small molecules that interfere with recognition or retrotranslocation of misfolded substrates. Overall, they managed to correct the activity of this misfolding, but have found that the ERAD inhibition is indirectly proportional to the loss of lysosomal activity, and that it produced varied levels of stress and unfolded protein response and induced apoptosis of these cells.
These are the references we got all our information from. Thank you very much for listening.